So this is not in your book. I'm just showing it to you so you can see it, to remind you that the endocrine cell makes a compound that it puts into the blood. The material, which we call a hormone, travels in the blood until it reaches target cells. What makes a target cell for a particular hormone responsive to that hormone? Receptors, good. And then the other kind of hormone is a neural hormone made by a neuroendocrine cell, which is really nerve tissue. A nerve cell makes a hormone that is dumped into a neurotransmitter onto
similar. So instead of releasing a substance into the blood, it releases the substance directly into a cleft right in front of the next neuron. So it's very similar. And we're going to be talking a bit about neurotransmitters and neurons and how similar they are in function to the neuroendocrine cell. Okay, the nervous system and the endocrine system talk to each other. And the nervous system actually has control over aspects of the endocrine system. And this is figure 1111 from your textbook. And you will see that the central nervous system controls a lot of hormone secretion. For instance, the hypothalamus tells a lot of different kinds of cells in the anterior pituitary lobe to make certain hormones. And we've already talked in detail about that. The posterior lobe of the pituitary is just neural tissue. It's the neural hypophysis. And so in that case, we have neuroendocrine cells making the hormones themselves. We also have information from the central nervous system talking to the interior of the adrenal gland, which I told you yesterday is more nervous than endocrine. And the adrenal medulla then is stimulated to release a hormone into the blood called epinephrine. Epinephrine also acts as a neurotransmitter between neurons. So there is a connection there. And finally, in this last picture, it shows a nerve coming from the central nervous system to a ganglion, a particular kind of ganglion, in this case in the autonomic nervous system. And this will make more sense to you hopefully after today's lecture, maybe after Monday or Tuesday's lecture. And it actually will stimulate a gland to make hormone. So these are various ways in which the central nervous system influences the endocrine system. Now, I want you all to just sort of shake and loosen up and relax. We are going to start the nervous system. The nervous system in your syllabus is supposed to take 2 lectures. I got news for you. It'll take us longer. Right, Scott? Yeah. Okay, so anyway, this is not a picture from your book, but using it didn't give me this over. Just to remind you that the nervous system has 2 sections. You have the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which is everything else. I don't know why I didn't illustrate. The previous edition of this book has in pale green and pale blue. This one is in gold and red. I think they're very hard to tell apart. But can you see that the spinal cord and the brain belong to the central nervous system? I will occasionally just say CNS for central nervous system because that's often what we call it. It's just CNS, central nervous system, and the PNS for peripheral nervous system.
neurons. This is a sort of a generic, generalized neuron from your book, 6-1. And the cell body in some situations is called soma. Even Vander occasionally calls it a soma, but I don't think Vander ever explains that it is using soma to mean cell body. So here someone's talking about the neurons. Soma provides an axon. What is the soma? Cell body, right. Okay, so the cell body has the nucleus, and the axon is the long piece of the neuron, long skinny piece of the neuron that leads to the next junction. And this particular so-called generic neuron is really an efferent neuron, E-F-F-E-R-E-N-T. It's what used to be called a motor neuron because it has an initial segment and the cell body at one end and axon terminals at the other. You'll see when I show you a picture of the afferent neuron how it's different. So dendrites come off of the cell body, and the axon is just a long extension with cytoplasm leading to a terminal portion of the neuron where interesting things happen, and I'm going to talk about those interesting things in just a little bit. Neurons can come with a covering or not, and in the peripheral nervous system, neurons have something called a myelin sheath, which is generated by Schwann cells. What Schwann cells do is they sacrifice themselves by wrapping themselves around an axon to insulate it, and we'll talk more about that later. This is 6-2A. In the central nervous system, there is also myelin. This is also 6.2. And in this case, the cells that create the myelin are called oligodendrocytes. They're different from the Schwann cells. So Schwann cells in the peripheral, oligodendrocytes in the central nervous system. And you can see that in each case, you have the membranes of the cells wrapping around. And this is also from your text, the same 6.2, and it shows in the central nervous system an axon surrounded by myelin. And each of these layers of the myelin sheath is actually membrane, double membrane of the cell. And one of the things that bothers me is that they had a great picture in previous Vanders, which they have ditched in favor of the ones I just showed you. But I'm going to show it to you anyway because I think it's really nice the way it shows you how the cytoplasm of the myelin-forming cell in the peripheral nervous system, the Schwann cell, is actually squished to the outside of the myelin sheath, and what we have inside is just membranes, because all the cytoplasm gets squished out from between the membranes, and you have just layer upon layer upon layer of phospholipids. What do you think is going to happen to the permeability of this axon if we have it covered with many layers of phospholipids? It's not going to be permeable to ions. That was interesting. Playing a piano. Okay. So what these cells do is they basically provide many layers of the membranes to insulate the axon so it doesn't leak. And we'll talk more about the function of the myelin in action potentials later. But this just shows you the myelin-forming cell starting. It looks more like a cell, then it starts to do this sort of spirally thing, and then it ends up being double or multiple layers of the myelin.
Question.
Yes, ma'am.
What's the reason our body doesn't work to produce all these layers if they're not required?
because we need to not have permeable places where they're not permeable ions. David?
Speeds up the transmission.
It actually myelinated axons are faster at transmitting action potentials than naked axons. So it actually speeds it up, and we'll talk about that in detail. Okay. So those, what I have, okay. When we're talking about myelinated neurons, we're often talking about, this is from another, this is the old term, motor and sensory neuron, because some afferent fibers have been found that aren't sensory, and some efferent fibers have been found that aren't motor related, but that's just why it says motor and sensory. So this is an efferent fiber and afferent fiber. So this is what we call motor, called efferent. Kind of hard to tell them apart when you say it, isn't it? Think the Latin word X, and think in terms of the central nervous system. Let's say you're in the central nervous system, and you want to go away from the central nervous system. You go through the door marked exit. Exit to leave, go out. And the other kind of fiber is called afferent, and that's from the Latin ad, meaning to, in this case, toward the central nervous system. So this takes messages away from the central nervous system, and the afferent fiber takes messages to the central nervous system. And look at the different shape of the afferent fiber. This is what you think of when you think of a neuron, isn't it? It's kind of the classic. But look at the cell body of the afferent fiber. It doesn't have dendrites on it, does it? It has instead what's called a peripheral process. Peripheral process. And in this picture it says dendrites. Well, you don't have dendrites on the peripheral process. I'm not sure why this author called them that. But this is 1987 is when this picture is made, so just bear with me. So no dendrites. Dendrites only on the cell body. And this is the peripheral process going to the peripheral aspect of the body, away from the nervous system. The part that carries the information from the periphery to the central nervous system. So it's to the central nervous system. This is called the central process. Makes sense? And to the central nervous system is the central process. And the cell body is off to the side. Just to show you the difference. Was that a question? Okay. In your book, you have 6.4, and it shows you the 3 different kinds of neurons and where you're going to find them. So the efferent fibers, some stimulate, others inhibit, but the neuron lies mostly in the central nervous system. You will see the cell body lives mostly in the central nervous system. The axon carries information from the central nervous system to outside. And in this example, they're showing the muscle gland or another neuron. The afferent fiber carries information from the periphery toward the central nervous system, and its cell body lives outside the central nervous system. So the afferent fiber has no dendrites in the cell body, and the cell body lives outside the central nervous system. Interneurons connect afferent and efferent neurons. That's what these are. This is interneuron. This is an interneuron. They live in the central nervous system, and they connect an afferent fiber and an efferent fiber. Interneurons account for 99% of all neurons. These little guys. I'm just counting neurons. Okay, in your book, you have a table 6.1, and it tells the distinctions between the three different kinds of neurons. I'm going to show it to you quickly. I don't want you to write this down. It's in your book. I don't want you to memorize it. Just understand that afferent fibers carry information to the central nervous system, but they live outside the nervous system mostly. Efferent fibers live mostly in the central nervous system and carry information out to away from the central nervous system, and also interneurons, which integrate signals between afferent and efferent fibers and live almost entirely within the central nervous system and account for most of the neurons. And I've given you a little relationship here. One afferent fiber has, for every one afferent fiber, we have 10 efferent fibers and 200,000 interneurons. So 200,000 interneurons for every one afferent fiber. You can see why interneurons make up 99% of all neurons. Yes?
Why do we need all those interneurons?
The interneurons are making the connections between afferent and efferent fibers, but they're also providing connections in your brain. So they're really important for connecting different things. I'm trying to think of a computer example. You know, if you have a peripheral thing like a disk drive, let's say you have a modern computer that doesn't have a disk drive, and you have this little diskette drive, and you plug it into that cord is like an interneuron. It's making connections between these two pieces. And if you don't have the cord, oh dear. Actually, most of the time now the cord is connected, but it used to be they were separate. So it's like we're making a connection. Okay, now you hold hands with her. Okay, so I'm a neuron, she's a neuron, you're a neuron, she's an interneuron. Okay? Raise your hand. What's your name?
Christina.
Christina is an interneuron. Okay. Did that answer your question, kind of, sort of?
Well, I'm still wondering why there's so many more.
Oh, so many more?
Yeah.
Because they are doing more than connecting afferent and efferent fibers. They're also connecting with each other. They're very prevalent in the central nervous. Well, they're mostly in the central nervous system, but there are a lot of them, and they are providing the bulk of the synapses in your central nervous system. Did you have a question?
Why do those afferent neurons use accents to receive stimuli? Because I thought only dendrites can receive stimuli from the outside.
Yeah, it's a terminology thing. Okay, Vander does not call those dendrites. He says that dendrites are just the processes on the cell body. Those are called sensory fibers by most people now. Okay, yeah, I know some people call them dendrites, but most. Do you think that everybody agrees in the scientific community? How many of you think that all scientists agree with each other? Raise your hand. Good. Nobody. Yeah, definitely. In fact, well, no, we won't get into that. That's a political statement about scientists, so I won't say it. Okay, so how am I going to do this? Let me just stop this. Oh! Can't you read backwards? Much better. Okay. There is another kind of cell called the glial cell. And the glial cells are accessory cells. And in the brain, glial cells outnumber neurons, including interneurons, about 1 to 9. So for every 1 neuron in the brain, you're going to have about 9 glial cells. And the glial cells come in 3 major flavors. And the first is oligodendrocytes, which we talked about. What do oligodendrocytes make? Myelin, right. Astrocytes Astrocytes are the utility outfielders. They do everything. They have many more jobs than I put here. They secrete growth factors. They actually guide axons in the fetus. They'll sort of say, come here, honey, keep growing, and they'll make sure that the axons connect to where they need to connect. They will clean up messes. They'll clear out cellular debris. They will push out an overabundance of certain kinds of ions. They secrete glucose and feed the neurons glucose, because neurons need to have glucose. They will not do well with fatty acids, or they can't do fatty acids or amino acids. They can only do glucose. Astrocytes just have many jobs. They also have slightly excitable membranes and can generate a small negative electrical potential and may be involved, we don't know how much, in sending messages in the brain. So astrocytes are pretty versatile. They do a lot of different things. What shape do you think an astrocyte is? Star shape. Oligos means many, or few, or a whole bunches, or none. What does it mean? It means a few. Yeah, so oligodendrocytes are cells that there are actually, they have, well, we won't get into the details, but they're not a whole bunch of those, but they're very important for myelin. Astrocytes are everywhere, and they do a whole lot of different things. Microglia, they're very tiny, and their job is as specialized phagocytes. They act almost like macrophages and eat bacteria and debris to get rid of it, to keep the central nervous system clean. They're the custodians of the central nervous system. And your book has a picture. And I got to tell you, there's no such thing as a green glial cell, but it's just to make it visible. So this is figure 6.6 from your book, and it just shows you the three kinds of glial cells. And what else do I have? Gesundheit. That was a good sneeze. Good sneeze. You don't want to just, you want to really let her rip. Okay, so neurons make about 10% of the cells in the central nervous system, but they make up about 50% of the volume. So they're only 10% of, if you count the cells, 10% will be neurons, but they make up 50% of the volume because they're larger than the glial cells. And one other thing, what does glial mean? Glue. So they sort of hold everything together. Okay. I'm only going to show this briefly, and we're going to go into this in much more detail later. This is 6.5. This is showing synapses between cells, because I'll occasionally be using that term before we get a chance to discuss it in detail. So the orange cell is synapsing on the green cell, and where it synapses is here it's synapsing on the dendrites of the cell body of the postsynaptic cell. This postsynaptic cell then can eventually be called a presynaptic cell because it also synapses on the next neuron, which is the next postsynaptic cell. So that's what this colorful picture is showing, is that synapses can occur sequentially. So one cell will synapse on the next cell will synapse on the following cell, and so forth and so on. So we'll talk a whole lot more about synapses when we get to that point. How are we doing time? This is always fun. Stand up. Rich, get the blood out of your butt and up into your head. Okay, you can sit down now. Thank you. It's almost time for our afternoon nap. That's I can see. You know, you should see what I see when I look out. At the beginning, I'll see people ready to start taking notes, and about halfway through, it's like, am I that boring? I'm sorry. Okay. Messaging in the endocrine system is chemical in nature. Messaging in the nervous system is electrical in nature. And the way we get electrical information passing through the nervous system is because there's a net charge on the inside of the cell that's slightly negative to the outside of the cell. And what this picture just shows you is a micro electrode inside the cell. You imagine that's a pretty small electrode, and then another one in the extracellular fluid. We designate arbitrarily the extracellular fluid as having a net charge or a total charge of 0. So whenever we're talking about negative and positive, we're talking about relative to 0 in the extracellular fluid. So the difference between the inside and the outside of the cell is called the resting membrane potential, or resting potential. It's called the resting membrane potential. And it's usually in most, not all, but most neurons about negative 70 millivolts. That's relative to an ECF, which is arbitrarily 0. So there's a small inclination for positive charges to leak into the cell and for negative charges to leave. However, how are those charges or those particles going to get through the membrane? What do we have to have there to get those things through? Channels and pumps, right. So we have to look at the channels and pumps on the membrane to see what really gets through. I don't think I'll do that yet. We'll do that in a minute. Okay, before we even talk about that, this is not in your book. I just wanted to show you. This is in the plasma. P is for plasma. I is for interstitial fluid. Intracellular fluid is inside the membrane. You notice that there's a whole lot more sodium outside the cell than inside, and a whole lot more potassium inside the cell than outside. Potassium and sodium are your major players. These other ions don't play a major role in action potentials. Some play a peripheral role, but they're not major. This is actually from chapter 4.1, and it shows the extracellular and intracellular concentrations of a number of substances. And I want to point out sodium and potassium, and actually you do have one in your book that just shows sodium, potassium, and chloride, I think. But sodium, lots more outside than inside. This is in millimolar amounts. Potassium, 5 to 150 outside. Chloride, you see that you have a lot more chloride in the interstitial fluids than you have inside the cell. And there are other things in the cell that help it be negative, slightly negative on the inside of the membrane. A lot of proteins provide negative charge. So these proteins will actually provide negative charge just inside the cell. When we're talking about resting potential of the cell, we're talking about just inside the membrane. The table you have in this chapter is 6.2 that shows the relative concentrations of sodium, chloride, and potassium.
Those are lipoproteins or transmembranes or both?
No, these are just proteins in the cytosol. Okay, so sodium is higher concentration outside, sodium and potassium is higher concentration inside. What do you suppose makes all that happen? Guess. The sodium-potassium pump. And this is a very old slide from Vander, 1998, the 7th edition. They got rid of this picture. Too bad. I like this picture. I'll show you what they've replaced it with. But I like it because it's so clean and clear, and you have the inside of the cell on the yellow side and the outside of the cell for the interstitial fluid here in the blue. And you have leak channels for potassium and leak channels for sodium, and then you have your sodium-potassium ATPase pump or antiport. Now, the thing that is deceptive about this picture is it looks like you have as much way for sodium to get in as potassium to get out. But the fact of the matter is there are many more potassium leak channels. Many more potassium leak channels than sodium. And of the sodium leak channels that there are, they are not as many of them open. There are not as large a proportion of the sodium leak channels open as there are potassium. So you have a net flow of potassium outside of the cell. And this is a picture from your current edition of Vander. This is 612. And this shows you the gradient. And you have potassium leaking out, able to leak out down its concentration gradient. And you have just a little bit of impetus for potassiums to go back in because of the electrical gradient. But the chemical gradient is very powerful and will make those potassiums leave. Sodium has a stronger driving force across the membrane because both the vectors go into the membrane. Both the electrical gradient and the chemical gradient push the sodiums into the cell. Okay, I want to make sure I cover everything. So the higher positive charge outside the cell actually opposes the potassiums leaving, but it still will leave because of the chemical gradient. Okay. So this is the generation of a diffusion potential. This is 610 from your book. And in this is artificial, but you can imagine you could apply this to a cell in interstitial fluid. Compartment 1 has a higher concentration of sodium, and compartment 2 has a higher concentration of potassium. So compartment 2 must be inside the cell. Good, thank you, David. And you can see that we're starting, in this experiment, we're starting with the same concentration. We have closed channels. If we open those leak channels, you will see that potassium is leaving a lot faster than sodium is leaving. And that's because sodium leak channels are usually either closed or there aren't very many of them to begin with. So in this case, we've got sodium leaving, and at a certain point, we get an accumulation of a positive charge outside that kind of resists the movement of did I say sodium? Mm-hmm. Call me on it. You don't know what it's like to be up here and trying to think and write and stand on your own 2 feet and talk at the same time. So I said sodium, I meant potassium. Potassium leaves the cell. At a certain point, the potassium carries enough positive charge outside of the cell that, or on the other side of this membrane, that we get a buildup of positive charge on the outside of the membrane and a negative on the inside of the membrane. So that then allows potassium to move back in through the leak channels. But look at the vectors. It's a very slight movement back in down the electrical gradient, but a much stronger tendency of the potassium to move out. Now, what this is a diagrammatic expression of equilibrium, but I think it's misleading. However, I'm pointing this out so that you understand that when the membrane leak channels are open, potassium will go out, mostly. Yes, there will be some leakage back in down the electrical gradient, but its driving force is outside. And we will show that in a little more detail in a bit. You guys awake? This is all really important stuff, because to understand the nervous system is important in order to understand the rest of the physiology, actually. So in this case, we're showing what happens when sodium channels are open and potassium channels, voltage, in this case, we will apply to voltage gated, not leak channels. So when the sodium channels open, we have the sodiums move in, and they actually create a more positive interior than we had when we began. This is 611.
That happened?
It happens during an action potential generation, yes. So I'm going to go back to 613. We're going to talk again about generation of the equilibrium potential of the membrane. This figure in your book has 3 compartments, and I'm not sure why they make it. I think they overworked the problem, personally. But the key here is the sodium-potassium pump. That's the key to just about everything. And it allows the cell to develop this gradient. The absence of gradients is death. And you've got to have gradients in order to be alive. And you can see for every 3 sodiums that are pumped out, the cell also will be able to pump in 2 potassiums. And they're showing leak channels. And you'll see that potassium is leaking out down its chemical gradient. As this pump is pumping potassiums in, potassiums are leaking out. See how that's a dotted line? And there's only one for these 2? That's the author's way of showing you there aren't as many sodium leak channels, and the sodium leak channels are not always open, so you get a lot less sodium leaking in. So you are actually establishing a net positive outside and a negative on the inside. So this is your resting potential. That's a steady resting potential. You have this pump continually hydrolyzing ATP to pump sodium out against its gradient and pump potassium in against its gradient. And then you have the leak channels operating. Whoa, slippery. Okay, there are 2 kinds of potential changes. Oh, I don't want to get into this yet. Need a blank. Well, that didn't get cleaned up very well. No, that's what you get for reusing. I like to recycle. So the membrane potential at which those 2 forces oppose each other, the electrical and the chemical, is called the equilibrium potential. This means there's no net movement or change of concentration of ion on either side of the membrane. The equilibrium potential is unique for each of the 2 ions, sodium and potassium. And the equilibrium potential is dependent on 2 things. 1, the concentration of ion in question. On either side of the membrane. In other words, inside or out. That determines the equilibrium potential. Also, the permeability of the membrane to each ion. The equilibrium potential of just potassium by itself is negative 90 millivolts. The equilibrium potential of potassium would also be the membrane potential if all of the sodium channels were closed and all of the potassium channels were open. But that's not the way it is physiologically. So let's take a look at how we get the membrane potential. The membrane were completely permeable to only sodium, then the equilibrium potential of sodium is equal to plus 60 millivolts. Do I want you to memorize these numbers? Thank you. No, I'm just doing this to derive it so you can see where we get the numbers. Because few sodium leak channels are open, and more potassium leak channels are open, and more potassium is leaving, the resting potential of a membrane is going to be closer to that of the equilibrium potential of potassium. The driving force of diffusion is going to be mostly chemical, and you're going to have a driving force of potassium outside the cell. That's what this negative means, going out of the cell, of minus 90, and of sodium plus 60, which means that it's the driving force in. The equilibrium potential of potassium minus the actual resting membrane potential gives you the driving force, actually. So let's say minus 90 millivolts minus the actual measured resting potential of the membrane will equal negative 20 millivolts. That's potassium out of the cell. So the driving force of potassium out of the cell is minus 20 millivolts, meaning 20 millivolts in the direction out of the cell. Now if we do that for sodium, we will have 60 millivolts minus 70 millivolts, just like we did with potassium. And that will equal, because negatives cancel each other out, 130 millivolts driving force of sodium in. So look at the difference. This is a much stronger driving force, 130 millivolts. So why doesn't the sodium rush into the cell given this driving force? That's not a rhetorical question. There's no way for the sodium to get in, right? The membrane is less permeable to sodium. Good. Okay, I don't have that one. Okay, so this is how we, yes?
Did you say that there are more of the channels for sodium?
Leak channels. Leak channels. There are fewer sodium leak channels, and of the sodium leak channels, a smaller proportion of those are open. So the membrane tends to be relatively impermeable to sodium, a little more permeable to potassium, which allows us to have the resting potential of the cell to be negative 70 millivolts. Okay. I'm going to write this down, and I want you to know this. That's a T. At resting potential, the cell is polarized. Because I'm going to use words and people are going to confuse them. They're going to say, what does polarized mean? Well, no, the cell's depolarized when it's resting. No, it's not. Polarized means that when you look at the charge inside and outside the cell, they're not the same. So you have a negative charge inside, a positive charge outside. So at resting state, the nerve cell is polarized. Changes in the membrane potential come in these types. You have depolarization. So when the membrane is depolarized, that means the membrane potential is getting what? More or less positive? More or less negative, I mean, inside the cell. Less, right. The membrane potential approaches 0. That doesn't look right. So it becomes less negative. That's depolarized. Okay, when it becomes hyperpolarized, does it become less or more negative? I'm hearing both, less and more. Oh, okay, thank you. Hyperpolarized, the membrane becomes more negative. Goes away from 0. Okay, I'm going to use a lot of times the term repolarized. And that's a return to the resting state that we're up here when we're polarized. Return to the resting. And then there's one final thing called overshoot. That means the inside of the cell becomes positive. Man, they're having...
So with overshoot,
the inside of the cell will actually become positive.
now these terms are going to come in handy when we talk about action potentials and graded potentials. But I think I'm going to wait till after the break to get started on those, because I can really see you guys flagging. I know it's Thursday. I know you think of it as the end of the week, but just bear with us for another hour, okay? One more hour. And this stuff is really exciting. So come back, please, at 10 after. Okay. There are 2 kinds of potentials. Only 2 you have to keep track of. And one kind is a graded potential. And this is 615 in your book. And the nature of a graded potential is that it doesn't go far. So here, let's say in the B section of this picture, let's say that we have a stimulus right here. This is where the initial depolarization occurs. And no matter whether it's high or low intensity, it will decrease in its intensity the further you get from the area where the initial depolarization happened. So you depolarize here, you have this response, and then as we get geographically removed from that spot, you will have a smaller depolarization. There are a number of reasons for that, but we'll get to that in a minute. So it can vary in strength depending on the stimulus. That's what this shows. It has a variable strength depending on the stimulus, and it is decremental, that is local, and the distance between, the farther distance between the polarizing event and where you are in the membrane, you will see a decrease. So it's decremental. And I'm going to write those words on an overhead in a minute. Okay, this is an old overhead from Vander. I actually like this picture, this diagram. They substituted that for it, the one I just showed you, but I like this one because it really shows the decay of the graded potential as you get further away. And the reason, you can see the reason. Let's say this is the interstitial fluid, this is the inside of the cell, and if you have depolarizing event, you will have positive charges coming into the cell and then moving in 3 dimensions out and into the screen along the membrane, pulling other positive charges to that point
And you will have this depolarization here, but as it moves further away from the point where you had that depolarization, you will have a decrease in membrane potential change. So this is just another way of looking at it, the same picture. So graded potentials have as their characteristics the fact that they are very local and short distance. They do not travel long distances. They go a short distance and they lose right. I thought that looked wrong. How many years have I been gone? 18 years of college and I still can't spell. Okay, they lose power the further they get from the point of depolarization. So they're very, very local. They don't travel great long distances. Also, they have another characteristic, which is why they're called graded, because they are graded, which means that the stimulus intensity determines, that's a T, not a P. The intensity of the graded potential. So the depolarization will be greater with a greater stimulus intensity. Also, here's another neat thing about graded potentials. Can be both depolarizing and hyperpolarizing. So some graded potentials have been known to actually make the cell more negative inside. Okay, now why do you think the charge gets less powerful the farther you get from the origin of the depolarization? That's number one. Why do you think? Anybody know? This is an old Vander. No, wait, this is in current Vander. 617. This is showing what happens during a graded potential. What happens is you have leakage of charge. So let's say we have a depolarization here, which lets in sodiums, and those sodiums travel along the membrane, but they can leak out as well as travel along the membrane. There's no new entrance of sodium because the sodium enters only at that point of initial depolarization. So eventually this charge or potential difference loses its power because it leaks. The charge leaks across the membrane. I also mentioned that it could be depolarizing and hyperpolarizing. And in that case, depending on what in the membrane at the site of the stimulus, what determines whether it's hyper or depolarization?
Positive or negative?
It's just the nature of the membrane proteins. Nature of the channels or transporters in that particular piece of membrane. And I'll give you detailed examples of that as we go along. Okay, also they are graded, so a weaker stimulus will make a smaller graded potential than a stronger stimulus. And this also shows you the whole thing about decremental. I think this is kind of confusing, but if the stimulus is here, and then we get that size of a graded potential, then I would say the stimulus here, and this would be, at a distance from this stimulus. It's just confusing to me. Another aspect of graded potentials is that there's no refractory period. That means, and you'll find out more about refractory period in a minute, that means that once you generate a graded potential, you can generate another graded potential and another right away. And in fact, because there is no refractory period, you can actually have what's called summation. So this is an example, this is an old version of 615. You can have one graded potential, or if you have several stimuli of that membrane, you could actually have 3 graded potentials on top of each other, leading to a greater depolarization. So that's what's called temporal summation, because it's more frequent stimulus of the membrane. This shows you spatial summation. That's where you have input from X, input from Y, input from both X and Y at the same time. But I'll show you a better picture of that will give you more details on that. But these are all features of graded potentials. So that other thing is that the graded potentials can be summed. So you can have summation with graded potentials. Also, no threshold. You do not have to reach or exceed a threshold for graded potential to happen. So what I mean by no threshold, this is an example. This is 621 in your book. These are graded potentials, and they can happen no matter how small the stimulus is. But an action potential has a threshold. And if you have stimuli that are sub-threshold, then you will not have an action potential. What has to happen is you have a stimulus that's large enough to elicit a graded potential that reaches threshold. Once that graded potential reaches threshold, then you can have an action potential. And even if you increase your stimulus, it doesn't matter, because once the action potential reaches threshold, it's going to, once the graded potential reaches threshold, it's going to turn into an action potential. Graded potentials don't have that. They don't require a threshold stimulus. They can be stimulated very little by a very small stimulus. So that's a difference between graded and action potentials. Okay. So the nature of the membrane tells you where a graded potential is going to occur. What kind of membrane do you think is going to have graded potentials or generate graded potentials? Say again? Well, sodium channels are often involved, yes. But I'm thinking of places where graded potentials would be. For instance, sensory receptors. Generate graded potentials. Also, the synapse that we've been talking about. You generate a graded potential at a synapse. You also generate a graded potential at the motor end plate, which will become more apparent when we talk about muscles. Motor end plate. That's a junction between a motor neuron and a muscle cell. And also, one other kind is pacemaker. Pacemaker potentials. So when you have a cell that makes its own, can change its own membrane potential spontaneously, can depolarize spontaneously with no outside intervention, That's called a pacemaker potential. Pacemaker potential very often is rhythmic. It will depolarize and the membrane will recover, and it'll depolarize and then the membrane will recover. Where do you find pacemaker potential? In the heart. That's a really good example. You also find it in other tissues, but in particular the ones you probably know about. One you know about is the pacemaker. So next we'll talk about the action potential. This,
Question?
yes.
Should we say that graded potentials have no threshold, or should we say that they haven't reached threshold yet? Once they reach
They require stimuli they don't require a certain stimuli strength. You can generate a graded potential with a very small stimulus. There is no threshold to generate the graded potentials. So graded potentials can be generated. Little ones, big ones, don't have a threshold of activity. Okay, so they don't have a threshold. However, I know where you're going with this. You can bring the cell up to threshold for an action potential. In fact, guess what the stimulus is for an action potential? It's a graded potential that has reached threshold. Okay, so they can provide the threshold for the action potential, but they themselves do not require a threshold to be met. So here we have the small stimuli generating graded potentials, but no action potential because we haven't reached the action potential threshold. And here is the threshold potential for action potentials, and you get an action potential. What do you notice as these stimuli increase? What do you notice about the action potentials relative to the stimuli? They're the same. So with an action potential, it's all or nothing. Either you get the action potential or you don't. So if you have a stimulus that's at threshold, you get the action potential. If you have a stimulus that's above threshold, you get the action potential, but it's not any bigger than the one at threshold. So the graded quality only lives in the graded potential. It's not something that is part of the action potential. The action potential, an action potential occurs only when sodium ions leave the cell faster. Enter spell checkers. Enter the cell faster than. Didn't anybody notice that?
Yeah.
Thank you. You should have hollered. Potassium ions leave. So we said that the leak channels allow potassium to leave all the time, right? In order to get an action potential, what you have to do is open enough sodium channels so enough sodiums enter to not only equal the number of potassiums leaving, but to be greater than the number of potassiums leaving. Because in order to get that positive or more positive charge, you need to have more sodiums coming in than potassiums leaving. So it occurs only when enough sodium channels are open to allow sodium entry to exceed potassium exit. Any membrane that can generate an action potential is called excitable, speaking of which. There's a great song in the 80s called Excitable Boy. Remember that one? Meatloaf, I think. Was it Meatloaf? Nobody knows that? Just playing. All right. So the action potential requires it requires threshold stimulus, unlike the graded potential. Unlike the graded potential, it's all or nothing. So you don't get a little graded potential, a little action potential and a big action potential. They're all the same size. Either you get one or you don't. The action potential has a refractory period. That is, right after an action potential, the membrane is not going to be inclined to make another action potential real soon. And we'll talk about the details of that in just a bit. I think that's all I want to say about that at the minute. I'm going to show you a couple more Vander slides before I write any more on that. Okay, this is a picture of the voltage-gated sodium and potassium channels, not leak channels. Do not confuse these with leak channels. Leak channels are different. Voltage-gated sodium channels and voltage-gated potassium channels respond to a change in membrane potential. And actually, they generate the action potential. So what happens is you have a closed sodium channel ready to open. You have a change in membrane potential, depolarization, that opens the sodium channel, and very quickly part of the protein blocks the channel so that no more sodiums can come in. That's called inactivation. And then it goes from inactivated state to closed state. After the sodium channels become inactivated, the depolarization opens the potassium channels. They open very slowly. One thing I don't like about this diagram is I'd like this potassium channel to be opening over here. It should be over here because it opens and closes quite slowly compared to the sodium channel. Gosh, we could be having, well, we are having fun, but in our own quiet way, right? Okay, this is 619. This diagram, I don't ask you to memorize much, but I want you to have this puppy memorized to the point where you can reproduce it. And the reason I'm saying that is to understand, to know this table and to understand, or this graph, and to understand it is to be able to talk all about action potentials and understand what's going on with action potentials. So I say don't memorize the tables, don't memorize numbers, don't memorize dates, but definitely know what's going on with this diagram, figure 619. On it you have a lot of stuff going on. It shows the shape of the action potential. And notice that the action potential, here we are at minus 70 millivolts, the resting potential. And at the point of stimulus, we have sodium channels opening. Now this is great because they're showing that the potassium channels are still closed when the sodium channels open. And you have this rushing in of sodium because of that driving force of 130 millivolts. And then at the apex of the action potential, you have inactivation of the sodium channels. And the potassium channels open. And what the potassium channels do is allow potassium to leave the cell so the cell can repolarize. Now the cell would repolarize because of that ATP using, oh, that's pathetic. The ATP using sodium, potassium pump. Okay, I want you to know that. But it would be a much more gradual repolarization. And we don't want this neuron to be out of commission after it fires an action potential for very long. So what happens is we have the inactivation of the sodium channel. No more sodiums are coming in. And the potassium channels open, so potassiums are leaving, and that makes for a sharper repolarization. This instead of repolarizing gradually, it repolarizes more rapidly. And actually what happens is it will hyperpolarize. It'll go a little lower than minus 70 millivolts. That's called the hyperpolarization. And at that point, the sodium channels are closed. They're not just inactivated, they're closed. And then we have both of them close at the return to resting potential. So action potential, repolarization, hyperpolarization, and return to the resting state. How come we go from hyperpolarization back to the resting state? What protein does that? ATP using sodium potassium antiport. Yeah. This is a nice little graph of relative membrane permeability for these two ions, sodium and potassium. And you notice that the permeability to sodium is approaching 0. And when you have your action potential, we have a very sudden increase in permeability to sodium. So sodium is rushing in, and then the sodium channel inactivates, and we have a sodium no longer permeable to the membrane as more and more of those sodium channels close. By the way, it's not just one channel, it's a ton of them. And then we have a return to our almost 0 permeability of sodium to the membrane. I want you to look at the permeability of sodium and the shape of the action potential. Isn't that interesting? So the shape of the action potential is very closely tied to the shape of this graph showing the permeability of sodium. This is overshoot, and the hyperpolarization is actually more tied to the repolarization is tied more to what's going on with potassium permeability. So I want you to spend lots of time with this. And when you start studying midterm, thank you, 25th, what's today, July 6th? When should you start studying for the midterm? Yesterday, yes, good answer. Or last week even. Studies have shown that if you wait until 2 days before the test, or the day before the test, or 10 o'clock at night before the test, you are not going to do as well, and you will certainly not remember as much a month down the line than if you study every day just a teeny bit. So the best thing you can do to prepare for this midterm is to go home, keep your reading current with the syllabus, even though we're a little bit behind, but keep up with the reading of the syllabus. Go home and look at your notes from today. Some people rewrite their notes. Some people have the most gorgeous notes. I'm embarrassed because my lecture notes are hideous compared to their gorgeous. I hear a lot of you typing your notes. I bet you have beautiful notes. So go home, edit your notes, clean them up, make sure that they are clear, because now is the best time to do that, not 2 weeks from now when you've forgotten everything we said today. And in doing that, that might take you, what, half an hour at the most? In keeping up with your notes, maybe even spending an hour more on reading or a couple more hours on reading, just keeping up with this fast-paced material, you will have more memorized and remembered by the time the midterm comes. Also, there is something in the textbook you really need to get into, and those are the thought questions, the applied questions, the ones that say, okay, a patient comes in, and these are the things that are happening with this patient. What do you think happened? Or what do you think will happen if we do this to this ATPase? What will happen if we poison the membrane with this substance that causes all the sodium channels to stop working? You need to do those types of questions, because that's my favorite type of question. I love questions that make you think and create something new. I don't want you to write definitions. You will not be writing definitions. I vow, I solemnly swear, you will not be writing definitions on this exam. You will not be well, you could label this graph. This one you should be prepared to label. You will not be recreating or spewing stuff that you've memorized that has no meaning to you. So what you have to do is find a way to find meaning. And sometimes that means you have to really work with the material. And what I always found useful to me was to do some kind of graphic organizer. So I would have a chart with, I would make my own charts, and it would help me organize my thoughts. Do not wait until the 23rd to start studying. If you do not have a learning community or a study buddy now, get one. And start talking about this stuff today. I think I made myself pretty clear. There's only one midterm. So if you want to do well on the midterm, do these things that I told you to do. Start studying a little bit every day. Look at your notes at the end of the lecture and edit them. Keep up with the reading. Get yourself a learning community now and start talking about these things today. Oh, and memorize this particular figure. Yes, sir, Matt.
What's the first word on the y-axis on the bottom graph?
The first word?
Yeah.
Relative.
Oh, I'm sorry.
Relative?
Yeah, it's easier to read this way, isn't it? Relative membrane permeability. This puppy is in your book. And I can tell you, this particular diagram, Vander has had since the first edition. They keep changing the diagram, but it's just one of the best. And it's nice to know what's going on with the membrane and what's going on with the action potential. This is a new piece that this edition has added, which is what's going on with the channels. Yes?
Do the two graphs match up like?
In time? Yes. Yes. So at 1 millisecond, this is one line going straight up. So this is occurring at the same time as this. This is occurring at the same time as this. So that's why I say the sodium permeability is a major player in the shape and the size of the action potential. So the shape and size of the action potential depends on how many voltage-gated sodium channels are in the membrane. Any other questions? I kind of got people's attention by talking about the midterm, didn't I? Okay. I cannot stress, overstress. I'm going to say it again. Don't stress. Yes, thank you. But also, start studying tonight if you haven't already started. Because this stuff, there's too much stuff to manage in one sit down. And I'll tell you something else. If you sit for an hour studying, that's it. You got to get up, go get a drink, go get a pizza, go call your friend, go walk around the block, play with the dog, do something, not studying. Interrupt that cycle. And then after you've had a chance to relax and reset yourself to a more comfortable state, go back to your study. And one reason you want to cut your anxiety is because, Gesundheit, as we will see when we talk about the senses, anxiety hormone epinephrine actually makes it harder for you to focus on print. So to read the print. So don't wait until you're anxious to read. Get comfortable. Have the fire burning in the fireplace if it's cold, or the AC on if it's hot. Have your dog sitting at your feet. Scratch his ears a couple times. Mellow, okay? Maybe a little glass of wine, not a big jug of beer, but a little glass of wine. Just get really mellow and then start reading. And don't say, well, I avoided reading these 15 chapters in all these weeks, and now I have to catch up on my reading tonight in an hour. No. Read every day while you're nice and relaxed. Sorry, I sound like I'm nagging, don't I? Well, I guess I am, kind of. But I think of it as giving you advice. Kindly, loving advice. Don't leave it to the last minute, because you'll be sorry. I know that that's the method, because I was an undergrad once too. And I remember. This is not in your book. This is an old Vander diagram that they've gotten rid of. It's a very fine diagram, I think. And what this shows is why the action potential does not lose its oomph as it goes from the cell body toward the axon terminus. So action potential that's propagated down an axon, even an axon as long as from the spinal cord of the giraffe down its shoulder to its hoof on the floor. You know, giraffes are tall, aren't they? The spinal cord about this high down to the foot. That's a long way for an action potential to travel, and it does not lose its power. Unlike graded potential, action potential will go all the way at full power. And the reason is because of the sodium channels, the voltage-gated sodium channels in the membrane. They will open in response to depolarization, and that will increase sodium permeability, which then causes depolarization of the membrane, which will open the next batch of sodium channels, which will then blah, blah, blah. What is this called? What kind of feedback? Positive. This is one of the few examples, yeah, it's written right there. One of the few examples of positive feedback that you will see in physiology is the propagation of an action potential by voltage-gated sodium channels. And I'm stressing voltage-gated because there are all kinds of sodium channels, but now we're talking about voltage-gated sodium channels, a very special breed of sodium channel. So what happens is the sodium channels open, they inactivate, they slam shut very quickly, but while they're open, there's an increased membrane permeability for sodium to flow into the cell, which then causes, it refreshes the depolarization. So as it moves down the axon, it is not going to lose its oomph. It's going to be recharged as it moves down the axon. So it's a very important aspect, because downstream, voltage-gated sodium channels will be open, and they will create their own action potential to pass down. Another feature I think I mentioned already of an action potential is, did I write that down? Yeah, I did write that down. But I didn't write that it's maximal. What that means is you don't get a small one. It's not graded. Well, I guess I kind of did with all or nothing. It's maximal, all or none. If you have a normal concentration of sodium in your extracellular fluid, you will have a maximal depolarization. But what I wanted to concentrate on here is the refractory period. People ask me, well, if the sodium channels are opening and causing sodium particles to enter the cell and move along the membrane in all directions, how come the action potential doesn't go back up toward the cell body? And the answer is the refractory period. So, is that right side up? Yeah, it is. Okay, so this is figure 622 from your, are you done? Okay. 622 from the book, and it shows that the current goes from the cell body toward the, in the case of an efferent fiber, toward the terminus. In the case of an afferent fiber, it would go from the peripheral process toward the central process toward the CNS. But in both cases, you've got unidirectional flow of the charges. So here's the initial site of the action potential, and this is showing you the graphing of the cell's potential. And you notice that downstream of where the action potential is occurring, you are having only a slight depolarization, and even further downstream, we're still at resting membrane potential. Then, as the current moves down, you will see that we are in the state of starting another action potential, and we're just starting to depolarize further downstream. But upstream, we've closed all of those sodium channels, and at this point, you cannot open them except with, well, actually at this point, you can't open them at all, because this is what's called the absolute refractory period. So that's absolute. And the reason for that is the potassium channels are still open, lots of potassium is leaving, and it's very hard to get enough sodium channels to reopen to let sodium in to counteract the flow of potassium out. So this is the absolute refractory period. And at that point, it doesn't matter how big the stimulus is, you aren't going to get another action potential. And then the flow of the current continues. And again, we see that we're starting a new action potential downstream. Here we are in refractory period, and here we've returned to the ability, we've reached this point where we're back to resting potential. Somewhere in this vicinity, you have what's called the relative refractory period. Gesundheit. And right about here is when enough potassium, voltage-gated potassium channels are closed that you might be able to stimulate enough sodium channels to open to counteract the flow of potassium outside the cell if you have a strong enough stimulus. So that's where you need a suprathreshold stimulus to get the action potential. Only during the relative refractory. So during the relative refractory, you can stimulate another action potential, but only with a suprathreshold stimulus. During the absolute refractory, no, you can't stimulate that membrane again. Yes, ma'am.
Well, is there a way that it brings it back to, I don't know, like even, I know the action potential won't be smaller, but will the next relative refractory be shorter or longer, or, you know?
No, it doesn't influence, the action potential is all or nothing.
Right. But the next relative refractory period will be the same.
Will be the same, yes. So once you have an action potential, the whole thing starts over again. And here's an old diagram from the old Vander. Slippery. Okay. This is also from the 7th edition, which, thank you, which I really like, but they don't have in the book anymore. Thank you. And it shows what's going on with the refractory period. So here we have a repolarized membrane in resting condition. This is actually the old version of one I just showed you. And what you have here is relative refractory period. That means that some of the potassium channels are closed. You now have less potassium leaving. It's possible to overcome the outflow of potassium with lots of sodium channels opening, but that takes a suprathreshold depolarization. During depolarization, you are in absolute refractory period. While the cell is depolarized, you're in absolute refractory period. While the cell is still positive, you are in absolute refractory period. So I just wanted you to understand that the refractory period is really important to keep the cell from sending the message in the opposite direction. So there is a table in your book, 6.5. This is an old version of it. It's really, really funky looking the way I have it, but just look in your books at 6.5. And it will help guide you in figuring out the differences between graded potentials and action potentials. And I'm not going to go over it right now because you guys, I believe, can read. So this is, I'm just pointing it out. I also wanted to remind you that there are, the only stimulus for an action potential is what? A graded potential, that is at threshold. Good. And the only stimulus for a graded potential, well, here they are. There are 3 of them, actually. Synaptic potential, which is a graded potential. Receptor potential, which is a graded potential. And pacemaker potential, which is also a graded potential. So synaptic potential means a potential that is generated at a synapse between 2 neurons. Receptor potential is a graded potential that's generated at a receptor for sensory energy like light, sound, temperature, that sort of thing. And the pacemaker potential, which happens spontaneously. And I have a picture, also from an old Vander, of pacemaker potential. This is very diagrammatic. It isn't what it really looks like when you do the tracing, but here we have an action potential. This is threshold, and this is 0. So it says that, resting 60. It's a little less than, a little higher than some membrane potentials. And you'll see this little green part that I've marked already, that's your pacemaker potential. That's the graded potential. So what this is, pacemaker potential, once we reach threshold, it becomes an action potential. It repolarizes, and then it starts the pacemaker potential again. The depolarization is spontaneous and usually rhythmic. We'll go back to pacemaker potential and see it in detail when we talk about the circulatory system. So, factors that influence an action potential. I'm going to have to use this mucky old thing. Sorry about that. Okay. Factors influencing an action potential. One is fiber diameter. That is the size, the diameter of the axon. The larger the fiber diameter, the less resistance to ion flow, and the faster the action potential. So a fiber diameter means a faster action potential. Another feature is whether the axon is myelinated or not. The myelinated axon has a faster action potential too. Also, because the myelinated axon has large stretches of membrane that contain no channels, and there's no reason for pumps, then we use less ATP. So myelinated axon uses less ATP and it's faster because of what I'm about to tell you. Oh, and also it takes up less room. If you have to have a larger diameter to speed up the action potential, then myelinating that fiber will allow the fiber to be narrower, smaller in diameter, and still have a very fast propagation of the action potential. So it takes up less room than a fiber that's not myelinated. This is from your book. This is 623. And this shows how myelinated fiber propagates an action potential. Between each of these, well, in the case of peripheral nervous system, Schwann cells, in the case of central nervous system, oligodendrocytes, you have these areas of myelination. These are all layers of phospholipids. You don't have anything passing in and out of the membrane there. But where you have no myelin, the nodes of, well, actually, some people call them nodes of Ranvier, but I've also heard them called nodes of Ranvier. So, I mean, it just depends on where you're studying it. But anyway, these nodes, you can call them nodes if you want, have lots and lots of ion channels. And this is where a lot of ions go in and out of the cell. And because of that, you actually have the changing, the current will travel from node to node to node. And this node hopping is called saltatory. From the Latin saltare, which means to jump. Yes. Okay. Nice sound, but I'd rather hear the Mac chime. That's my favorite. Okay, so the myelin insulates the neuron. Fewer sodium channels in the membrane covered by myelin, which makes sense. Why should there be any sodium channels in the myelin covered by myelin, in the area covered by myelin? It excludes water, soluble ions, and prevents leakage. The nodes are very permeable to sodium and potassium, so the impulse then can hop along more quickly than if it had to go along opening channels as it went. I hear some whispering. Is there a problem? Is there a question? I want to make sure that, am I over time? Oh, it's 4 o'clock. Okay, so that's where we'll start on Tuesday. Guess what you have to do this weekend? You're going to study for a midterm every day. Oh! Before you go anywhere, we have the quizzes to hand back. If you have a question about the quiz, don't talk to me. Talk to Michael. And what he's going to do... Oh, I'm sorry. And also, turn on the light there. Near Meet is also helping. It has office hours Thursday 1 to 2. Put your hand up here. Upstairs, 307.